In part 2 of the face coding lesson, we'll try to get a feel for how neural activity might code for a face in a face region. Historically, there have been two views about this, the cluster view and the distributed view. In the cluster view, it's relatively few neurons that respond selectively to each face, representing identity. A cell's preferred stimulus is a unique face. So here we see the fusiform face area, and the idea is that there are lots of cells in the fusiform face area, but only a relatively few neurons selectively respond to any given face. So here we have the red is going to be responding to face A, the yellow responds to face B. We have the response profile here, and we see that neuron number 1 responds a lot to face A but not face B. So does this neuron. But neuron number 4 has the reverse pattern of activation. And notice, out of all the neurons in the region, it's a relatively small number of neurons that are selectively responding to unique faces. So this is called sparse coding. That it's only a few neurons that are selective to a given face. And they're very selective. Now that's in contrast to the distributed view, where lots of cells are responding to faces. So in this view, many neurons respond to both faces, but the overall pattern distinguishes the faces. A cell's preferred stimulus is a facial dimension. So here we see that a lot more of the cells are responding to both faces, but they don't do so equally. Here we have a neuron that responds more to face B than to face A. Here we have a neuron that responds more to face A than to face B, but it's the overall population pattern of activity that would be able to code for the unique identity of a face. And this is called population coding. Now, for both of these views, it is true that perceptions and long-term memories of a face involve activations of populations of cells with preferred stimuli. In the case of the sparse coding, the preferred stimulus is a unique face. In the case of population coding, the preferred stimulus might be just some facial dimension, a more narrow aspect of a face, like how wide is the face, how the distance between the eyes, the hairline, etc. To better understand the difference between these two views, let's look at a simple picture like this. Here we are in the fusiform face area. These cells are receiving visual inputs from earlier visual pathways, and we have 3 cells that are shown to be making action potentials, the little red marks here. And this would be the sparse coding view. So face number 1 has activated these 3 cells. So it's a minority of cells. So this is sparse coding, and they are very face selective because, as we'll see in the next slide, these cells do not respond to face number 2. It's a different population of cells that respond to face number 2. Now, how did these cells come to respond to their respective faces? Well, it might be that during face learning, there's LTP going on, synaptic changes are creating these target cells with their preferred stimuli. But in this cluster view, the reason why a different face doesn't activate it is because it's a different face with a different combination of characteristics, a different identity. So in the sparse coding view, these cells have a preferred stimulus of a unique face. Now, in the distributed view that uses population coding, here face number 1, you'll notice, is activating a larger number of cells in the population. So it's not sparse coding. It's sort of a population coding. It's a distributed form of representation here. And a given face cell might respond to multiple faces if those faces have some shared characteristic, like distance between the eyes or the width of the face. So let's focus on this cell here. It did respond to face number 1, making lots of action potentials. And here comes face number 2. That cell is also active. How would we explain this? That this cell is not coding for identities or unique faces per se. It's focused more narrowly, maybe just on the distance between the eyes. And so 2 very different faces might have the same eye distance and so activate this cell to the same degree. So according to the distributed view, face cells are face selective because they are responding to faces, but their preferred stimulus is not a face per se, but one characteristic of a face that may be shared or not with other faces. Take this cell here, responds more to face 1 than to face 2, but it does respond somewhat to face 2. So maybe this is a different dimension of a face, the width of the face. Maybe the preferred stimulus here is a wide face, and that maximally activates it, while a narrow face will not activate that cell as much. So in the distributed view, the memory for a unique face involves a unique activation pattern in cells responding to face dimensions. Each face has a unique set of characteristics, so a population of cells will have a unique activation pattern. We've already seen that these two cells here might respond similarly if the faces have the same distance between the eyes. Here we have maybe the different width of the faces is shown up in the differential activation here, and maybe these cells are responding to some other dimension. So face number 2 activates this cell a little bit, but face number 1 does not. As it turns out, some recent experimental results seem to support the distributed view as against the clustered view. And the paper was called The Code for Facial Identity in Primate Brain. And the researchers recorded from lots of cells in these face selective regions of the brain. So let's think about faces again. So here are some faces, and they differ along many dimensions: the distance between the eyes, the width, the hairline, etc. Also just sort of skin tone and other kinds of lots of features that might distinguish faces. And one of the ideas is that face cells maybe aren't responding to unique faces, as in the sparse coding view, but maybe they're tracking facial dimensions with a preferred stimulus along one of the many facial dimensions. So for instance, a given cell might respond maximally when the distance between the eyes is large rather than small. So what the researchers found was that some of these face cells responded to very different faces, so long as the faces shared the feature along its preferred dimension. So let's just pretend these 4 very different looking faces all had the same distance between the eyes. See, then what they were finding was some of these face cells were responding equally to these very different faces. So that does not support the sparse coding view. And then they went on to characterize these face cells. What dimension were they responding to? And based on face cell activity, then the researchers could predict the face that the animal was seeing. So this was the actual face. They recorded from cells. They knew the response characteristics of the cells, and without actually even knowing what face was being presented, they could predict the face. They could reconstruct the face based on the pattern of cell activity, and the prediction was pretty close to the actual face that was presented to the animal. And interestingly, it was just around 200 neurons were sufficient to uniquely characterize a face. Now that starts to sound like sparse coding, but remember, in the sparse coding view, the cells were responding to the whole face, in a sense, the identity of the face. These cells are responding to face dimensions, but it wouldn't take that many cells. The suggestion here is it wouldn't take that many cells to have a population of cell activity coding for unique faces. And so the most recent understanding then is that the face areas contain face cells, but these face cells are tuned to kind of narrow facial dimensions. And it's the pattern of activity in the population of cells that are responding to faces that identifies one face rather than another.